On October 5th, 2020, the 2020 Nobel Prize in Physiology or Medicine was awarded jointly to three scientists, Harvey J. Alter, Michael Houghton, and Charles M. Rice, who made crucial contributions to the discovery of the hepatitis C virus. For more information, we have Dean Tran at the scene.
Thank you, Sian. Yes, as you mentioned, this recent Nobel Prize was awarded to three research pioneers, Dr. Alter, Dr. Houghton, and Dr. Rice, all of whom helped discover the hepatitis C virus. The word hepatitis is inflammation of the liver. Hepat means liver and itis means inflammation, and is commonly caused by infections such as the hepatitis A, B, and C viruses. However, there are many causes of hepatitis, and heavy alcohol use, obesity, toxins, certain drugs and medications, and other medical conditions can also help give rise to hepatitis. Before hepatitis C was discovered, two viruses were known to cause hepatitis: the hepatitis A virus, or HAV, which is very contagious and spreads through close contact or contaminated food and drinks, and the hepatitis B virus, or HBV, which was the only known blood-borne pathogen causing hepatitis. However, in the 1960s, Harvey J. Alter showed that there were many patients with a form of blood-borne hepatitis that could not be explained by HAV or HBV. Thus, the search to identify the mysterious pathogen causing non-A, non-B hepatitis began.
For more than a decade, the virus for this mysterious form of hepatitis evaded traditional ways of identifying viruses. However, Michael Houghton tried a different method that proved to be fruitful. After isolating DNA fragments from the blood of a chimpanzee infected with this mysterious virus, Houghton tried to isolate the DNA generated from the virus from the rest of the chimpanzee DNA. To do this, he inserted these DNA fragments from the infected chimp into bacteria. DNA is the recipe for making proteins, and so these bacteria read the DNA recipe and created proteins as instructed. Next, Houghton took the serum from the blood of non-A, non-B hepatitis patients, which contained special proteins called antibodies that are like keys that fit in and stick to certain proteins from the virus. So, these antibodies stuck to bacteria that contained the viral proteins and helped Houghton isolate the DNA from this mysterious virus. Further research by Houghton and other scientists showed that this virus was an RNA virus, and they named it hepatitis C. RNA is a chemical similar to DNA and is used by many viruses such as hepatitis C, HIV, and COVID-19 as their recipe instead of DNA.
Although Houghton's discovery was monumental, scientists still had to confirm that this was indeed the mystery virus that caused non-A, non-B hepatitis. In other words, researchers had to show that this virus alone was able to replicate and cause the disease. For this task, Charles M. Rice, along with other researchers working with RNA viruses, discovered a region at the end of the viral genome that may be important for viral replication. To test this hypothesis, Rice genetically engineered an RNA variant of the hepatitis C virus that included this viral replication region. After injecting this RNA into chimpanzees, the virus was detected in their blood and the chimpanzees developed pathological changes that resembled hepatitis in humans. Thus, Rice's research showed that hepatitis C virus by itself could explain the mysterious blood-borne hepatitis.
Thanks to these discoveries, highly sensitive blood tests and antiviral drugs for hepatitis C have been developed. As a result, hepatitis C is now curable over 95% of the time, and there are elimination efforts occurring across the world. Hepatitis C has the potential to be an infection that can be eliminated without an effective vaccine. For the future, our next step is to carry out international efforts to make blood tests and antiviral drugs widely available in hopes to someday eradicate hepatitis C from the world population. To learn more about one of these efforts, visit the World Hepatitis Alliance website in the link provided in this video's description.